Thanks, Luna. Yeah, so I'm Michael Lazear, and I work at Belharra Therapeutics. And one of the projects I've been working on is a proteomics search pipeline called SAGE. At Belharra Therapeutics, we leverage mass spec-based proteomics to discover drugs for undruggable proteins. What this means in practice is that we synthesize a library of diverse chemical probes that capture proteins they bind to. We can then extract these proteins from the sample, sequence and quantify them, and this essentially allows us to reverse the traditional drug discovery approach. Whereas you might typically screen millions of compounds against one protein target, we instead screen thousands of diverse compounds against thousands of targets in parallel to discover new drug-protein interactions. So I say mass spec-based proteomics, but what does that really mean? If you're not familiar with them, proteins are the molecules that carry out life in biological processes. As such, they're the molecular targets of 90% of FDA-approved medicines. You can think of proteins as strings of peptides and peptides as strings of amino acids. And amino acids are a set of 20 commonly found chemical molecules that make up all peptides and proteins. And they have this alphabet here. Just have some examples like Leslie K, edit pep, etc. So each of these 20 amino acids has a unique chemical structure and molecular mass. And in peptides, the chemical bonds that chain these amino acids together fragment in a predictable manner, which is pretty useful if you want to sequence something via mass spectrometry. So I just have here a very simple chemical diagram of what a peptide backbone might look like, as well as some of these different fragment ions, as we call them. And proteomics typically focuses on this B and Y series ions. And you can think of these as basically prefixes or suffixes. So for instance, if we had this peptide Leslie K, we would have this series of prefix B ions, LES, LESL, LESLI, etc., and the set of Y or suffix ions shown here. In a typical proteomics experiment, we want to leverage this information about those predictable fragmentation patterns to sequence proteins in a sample. So we take our proteins, we chop them up with an enzyme into small peptide sequences. So again, these are strings of amino acids, but now they're small enough that we can actually shoot them into a mass spectrometer and measure their mass over charge. However, proteomic samples typically are very complex, where we might have 5,000, 10,000, 15,000 proteins in a single sample, and hundreds of thousands or even millions of unique peptide sequences in a given run. So we separate them using liquid chromatography, basically uses chemical properties of peptides to separate them over the time dimension. Then within each full mass spec scan, we basically can read out the mass of individual peptides. And that correlates basically to the sum of their amino acid masses. We then select one of these peaks for further fragmentation, where we can read out these prefix and suffix masses, those B and Y ions. Basically, with these two pieces of information, they're sufficient to actually identify peptides. And just very briefly, this is an example of what a real mass spectra might look like with some peptide sequences assigned to the top and bottom. So two different sequences with their prefix and suffix ion. So it's kind of complex. You might end up with multiple peptides per spectra. But we also kind of have a problem. Most existing proteomics software tools are closed source and flow. And as you might expect based on the previous couple slides, proteomics is totally dependent on software to annotate and quantify proteins. That involves trusting closed source and proprietary software. And modern mass specs are getting faster and faster, generating millions of these spectra and hundreds of gigabytes of data per day. So kind of the existing state-of-the-art tool and previously fastest tool is called FragPipe, an academic tool, but it's closed source and non-free, which is pretty typical for proteomics for whatever reason. Instead, I developed a version of this algorithm in Rust. It's MIT licensed, free for everybody. And we get about a 10 to 25x speed up over the current kind of existing state-of-the-art tools. Basically, we do this by, proteomics is essentially an embarrassingly parallel problem where every stage of this pipeline can basically be done in parallel, but existing tools are kind of embarrassingly single-threaded. So instead, extensive use of Rayon along with some clever algorithms and data structures allows us to really speed up this problem. So a traditional proteomic search might involve taking this precursor mass along with a mass error window, since our instruments are not, super precise. I mean, they're very good. You can get within, parts per million accuracy, but you still need to look for multiple peptides per spectra. Traditionally, you might look at every peptide that matches this mass and window and compare each of their spectra one by one. Instead, Sage and FragPipe before it use this kind of fragment index, essentially a B+ tree, where the outer layer of the tree is fragment masses and the inner layer of the tree is precursor masses. This allows us to basically search fragment peaks one by one to see if they match up to a prefix or suffix mass. We can then associate that immediately with a peptide and a precursor mass. This gives us 100x speed up over traditional tools, and the use of Rust gives us another 25x speed up. Basically, we can search 650 gigabytes of data in 12 minutes. Sage is also the first proteomics tool that enables direct kind of streaming and decompression in RAM from S3. So we store all of our data in the cloud. We can basically transfer those compressed files directly into RAM, search them, perform probabilistic rescoring and false discovery rate control. And we actually finish before the previously fastest tool can even begin running. So we have to download and decompress those files onto an NVMe drive before searching with traditional tools. So despite using no disk IO at all, we still use less RAM. It's way cheaper, way faster, and it's open source. Quite nicely, we get about 98.2% overlap with our spectra identifications. Not only can Sage sequence peptides faster, but we also do it more accurately and precisely than existing closed source tools. So we can use a ground truth experiment where we spike E. coli peptides into cancer cell lysates at known ratios. Sage accurately recapitulates these known ratios and does so while identifying more proteins than our competitive tools. And we also do so with higher precision as measured by coefficient of variation of different peptide measurements. I know I kind of blew through this, but in conclusion, Sage enables fast, scalable, and cost-effective proteomics searching in the cloud. It represents kind of a step change in speed and scalability of proteomics toolings where it's feasible to search thousands of files at once now. You can scales up to terabytes of data in minutes. Quite nicely, the ability to stream from S3 really unlocks limitless storage and compute. Rust is just a phenomenal language, super easy to install, static Rust binaries from Docker packages, conda distributions, etc. And most importantly, this just represents more open source software for biology and drug discovery. Thank you for your attention.